Hi, it's Dr. Ogden again. So in the last lecture we saw kind of an overview of some of the major events leading up to the diversity of life on this planet. And we're going to kind of pick it up from there as we look at the three main domains: bacteria, archaea, and the eukarya. The bacteria and archaea belong to are both constructed of prokaryotic cells, and so that's why we have this prokaryotes here in this bracket. But it turns out that archaea are actually more closely related to eukarya than they are to the bacteria. Now, we're just going to quickly look at some examples of both bacteria and archaea, and then we'll spend more time looking at the eukaryotes. So here is just three examples of the diversity that you can see in the different shapes of prokaryotic cells. Sometimes the cells can be spherical. These are called cocci, or you can have rod-shaped cells like the bacilli, or you can have spiral-shaped cells. And each of these different forms serves different purposes in the environments in which they live, and they've adapted these different forms to be able to survive better. Also, some of these prokaryotes, like these archaeal karyotes here, they prefer environments that most other organisms don't want to live in. So there are some archaea that are salt-loving and some archaea that are heat-loving, like those that live in the hot springs of Yellowstone. But the real interesting question, I think, is what happened along this branch of evolution that got you from a prokaryotic cell to a eukaryotic form of a cell? And then how did that diversify into all of the different lineages of eukaryotes? And this idea is actually well studied, and we have a very strong theory about how this occurred. And remember, theory in science is as good as it gets. And so what we accept that happened is that from an ancestral prokaryotic cell that had just DNA, right, and no membrane around that DNA, so there was no nucleus, you began to have plasma membrane infolding. And so these infoldings began to form the endomembrane system within the cell. And so eventually you get membranes that surround the DNA to form the nucleus, and you get the rest of the membranes that start to form the other organelles in the cell, like endoplasmic reticulum, Golgi apparatus, and so forth. Then the other important part in the step of leading to the eukaryotic cells is that there were these relationships that were formed between aerobic heterotrophic prokaryotes. So this aerobic heterotrophic prokaryote comes inside through the process of endosymbiosis, and you now have this relationship where one cell is living inside of another cell. Endo inside, symbiosis, meaning they both help each other out. And it turns out that an aerobic heterotrophic prokaryote is very good at making ATP, making energy. And on the other hand, this cell then did not have to spend as much time making energy. It could spend time doing other things. And what you'll see is eventually this then continues to evolve and continues to evolve and eventually becomes what we call a mitochondria, around the similar time in other lineages of these early eukaryotic cells, some eukaryotes also brought in photosynthetic prokaryotic cells and started also an endosymbiotic relationship with these. And then these led to what is now we call chloroplasts, which are found like in plants, right? These are the things that perform photosynthesis inside of plants. So you had a multitude of different cells exploring these different pathways, and eventually some of these endosymbiotic relationships led to mitochondria and some of them led to chloroplasts. Now, is there any evidence to support this? Because that sounds like a real great story, but as I said, this is a theory, so it's really strong. It's our best idea based on all of the evidence. So what is the evidence? Well, first of all, mitochondria and chloroplasts can only arise from existing mitochondria and chloroplasts. So you don't make mitochondria and chloroplasts from something that's inside the DNA and the nucleus. They can only come from pre-existing mitochondria and chloroplasts. Now, they also have their own genomes. And so they resemble, and those genomes resemble those of prokaryotes. They're circular, not linear, and they're about the same size. And so this is what's really interesting, is that when we talk about all of our DNA, we have DNA inside of our cells, right? Remember, we've even mentioned this before, 46 chromosomes. But we also have another chromosome inside of our bodies, inside of each of our cells, and that's found in the mitochondria. Plants have their nuclear chromosomes, then they have the mitochondrial chromosome, and then they also have chloroplast DNA. So they actually have 3 areas where DNA is stored. Another piece of evidence is that both mitochondria and chloroplasts have their own protein synthesizing machinery. So they go through the process of protein synthesis just as happens in the nucleus, and as the nucleus sends out the messenger RNA, and then that goes through the process of translation to make proteins. That same similar process is all found within the mitochondria and chloroplasts as well. And then, they're just similar in size. So there's lots of evidence that way. But we can actually empirically test this idea. And to empirically test this, the way that you do this is you do a phylogenetic analysis. So once again, phylogeny really informs us about evolution. And so the way this particular example is showing all of the genes that have been sequenced, so all of the genetic information for a bunch of different archaeal species, a bunch of different bacteria species, and then some eukaryotes up here. And here we are, right? Homo sapiens, right next to Xenipus laevis, and I don't know if you know what that is, it's a frog. Maize, if you speak Spanish, maíz, that's corn, and then a bunch of other things. Most of these are protists, right? And so what you do is if you sequence all of the genomes from all of those organisms, this is the tree that you get. And then if you sequence the genomes of chloroplast and mitochondria, they actually nest over here being closely related. So here's maize mitochondria. So the mitochondria from corn nests right over here, right next to this agrobacterium. And then the chloroplasts nest right within this group of cyanobacteria. So this supports the idea that way back in time, mitochondria were once bacteria, and chloroplasts were once bacteria. And that over the millions of years of having that endosymbiotic relationship with those early eukaryotic cells, they basically continued to evolve and basically became an organelle, but it's an organelle that still has DNA. And when you look at essentially the fossil history of that DNA, it shows that it's still really bacterial DNA. It's not eukaryotic DNA. Because if it would have just been DNA that somehow got out of the nucleus and over into the mitochondria, you would still expect then both the corn mitochondria and corn chloroplast to be right over here next to the corn, but it's not. It's over here by the bacteria.